Hello, welcome back to another video, Biology of Cells and Tissues. Continuing with the nervous tissue, the nervous system tissues, we're going to go right into the glial cells. Of course, if you can, leave a like on the video, subscribe if you're not already. We're almost this close to hitting the 500 subscriber mark. Helps me out a ton. So, talking about the glial cells. Glial cells, these are our supporting cells. They're also called neuroglial cells. Specifically, we're talking about them in the context of the nervous system. They are the supporting cells. So while the neurons do all of like the message transport, these cells are kind of running the rest of the show, making sure that the neurons themselves are able to do their job. So we're going to split it in between two groups. We have the central nervous system glial cells and then the peripheral nervous system glial cells. We'll talk about the CNS first, then we'll talk about the PNS as well. As well, when we get through all this, these are the ones that we're going to talk about: ependymal, microglia, astrocytes, oligodendrocytes, Schwann cells, and satellite cells. As we go through this, there is going to be a little bit of jumping around because there is a little bit of overlap as far as function goes. So the oligodendrocytes and the Schwann cells both do a similar job, but they are very, very different from each other. So we're going to get to all of that towards the end as well. So just kind of, you'll see, and I'll point it out, like, hey, we're going to talk very briefly about this, but then we'll go over it again later on. So glial cells of the central nervous system. Here's a nice little picture of all of these different cells that we're going to be talking about: the oligodendrocytes, the ependymal cells, astrocytes, and microglia. These are the big ones that we're going to talk about. So starting with the astrocytes, these are our workhorses. There are tons and tons and tons of them, and they take care of a lot of the general jobs that need to be done. Biggest one, the one that's most commonly asked on exams, is that they control the blood-brain barrier, meaning that they make sure that blood isn't leaking out into the actual brain tissue or into the brain space, because if you increase pressure inside of the cranium, inside of the cranial cavity, that puts pressure on the brain, and that's a very, very bad situation. So the blood-brain barrier, its other job is also to protect the brain, make sure that only the specific things that are allowed into the brain that the brain needs, like its nutrients, oxygen, blood, that kind of stuff, make sure that only those things come through. Okay, that's the big job of the astrocytes. They also respond to injury, formation of scar tissue. Now again, the neurons themselves are not able to replicate, so depending on the type of damage, we'll still have issues with that, but the astrocytes are able to protect those specific areas and lay down scar tissue when necessary. There's two specific types that we mention. We have protoplasmic astrocytes, which are primarily found in the gray matter, and they have short, thicker processes like this, and has a ton of them. So lots and lots and lots of these. These are in the gray matter. Then we have the fibrous astrocytes, which are much longer and thinner processes, all reaching out, and those are found in the white matter. This is more just, recognize the names, and it's not super, super important, all of the details. Just the name, kind of the location, this little processes. Both of them are doing the same types of work. Here's just some of the more histological picture of it. Here are astrocytes right there surrounding the blood vessel, again, because they're part of that blood-brain barrier, making sure that only the specific things that are allowed to come through come through. And we see the black staining as well. A lot of our glial cells, we use that silver stain in order to see those properly. The next glial cell, or neuroglial cell that we're going to talk about, are oligodendrocytes. So the job of the oligodendrocytes, they are responsible for creating and maintaining the myelin sheaths. We talked a little bit about myelin sheaths already, just briefly. We talked about the anatomy of the neuron, but we're going to get deeper into the myelin sheaths and how those are all created and kind of really what they do in this chunk of the material. The main job of the myelin sheath, it acts as an insulator. This is almost always a test question, that its job is to insulate, to allow messages to be transmitted much faster. This is the big important thing about oligodendrocytes. A single oligodendrocyte attaches and produces myelin for multiple axons. So we'll see one single oligodendrocyte that reaches out, it's like here, it reaches out and acts on multiple different neurons. It lays down several internodes, we call these internodes, of the myelin sheath. And that again is in the central nervous system. It's always asked on tests about the myelin, comparing central nervous system to peripheral nervous system. So make sure that you know central nervous system, oligodendrocytes, and they produce myelin for multiple axons. Always test question. Next, we have the microglia. These are our immune cells derived from monocytes. We talked about monocytes a couple of times. Remember, monocytes turn into the macrophages. So microglia are the macrophages of the central nervous system. Okay, they go, and if there are microorganisms, cellular debris, anything that's not supposed to be there, or it's not functioning properly, the microglia are going to come, munch it all down, digest it, and get rid of it. And these microglia especially line what are called the ventricles of the brain, as well as the spinal cord. Because our spinal fluid, it's called our CSF, our cerebrospinal fluid, is often where we're going to get stuff that we don't necessarily, if there's going to be a place where there's stuff that we don't want it to be, it's typically going to be inside of that fluid. So they line around that fluid, and they make sure in order to capture anything that's in that fluid that needs to be taken out. The cerebrospinal fluid is also kind of responsible for some of the waste carrying away. So it makes sure to help get rid of the waste, and especially when we sleep. So there's a technique in chiropractic called SOT, sacral occipital therapy. I believe, pretty sure it's sacral occipital therapy. They have a theory in that technique which talks about the pump of the cerebrospinal fluid, how the cerebrospinal fluid has a specific flow, and a lot of times what we would refer to as subluxations can mess with that specific flow, which then affects the rest of the body. So it's a cool technique. It's interesting. So definitely, when you get to a point in your journey, you can check that out. Again, so microglia are our immune cells. Finally, continuing to talk a little bit about that cerebrospinal fluid, we have ependymal cells. So ependymal cells, these are simple cuboidal or columnar epithelium. Remember the epithelium, the different shapes of the epithelium. They're often ciliated. Remember, cilia are little hairs. They have the actin and stuff inside of them in order to kind of brush, to pump, in order to move stuff around. The main function of these ependymal cells, they deal with the CSF, the cerebral spinal fluid. They do secretion, maintenance, and circulation of that spinal fluid. And again, where's that spinal fluid found? Inside of the ventricles of the brain, which we'll look at here in a sec, as well as the spinal cord. Remember when we talked about the spinal cord, you had that central canal. That central canal, that's part of where either you have cerebrospinal fluid going through there, as well as all along the outside, inside of what's called the dura mater. You should learn all about that in anatomy, as well as your neuroanatomy courses down the road. I want to find real quick, when we look at pictures of the brain, these holes that we have, these are our ventricles. They're essentially these spaces inside of the brain in order to deliver some of the nutrients, some of the other stuff that we need, take away the waste from the inside. We need those spaces to inside of the brain as well. The ventricles themselves, you'll learn a lot more about the ventricles in neuroanatomy. You'll learn about kind of what they are, what they like, the flow of the CSF, but in all of the ventricles, as well as kind of the development of those. That's a concept that's often tested on Part 1 boards, so you'll see that in the future. And hopefully in my videos, once I finally get through material and I can start producing Part 1 board material. So we mentioned that the ependymal cells are a component of what's called a choroid plexus. The choroid plexus is inside of some of those ventricles in a couple different locations. And here's the spaces. There's a ventricle, another ventricle, and this is called the cerebral aqueduct, a fourth ventricle down here, and all the way down. So the choroid plexus, or these ependymal cells, line the inside of those ventricles. And that is the central nervous system glial cells. Moving on to the peripheral nervous system glial cells, we have two that we want to talk about: satellite cells and Schwann cells. We mentioned satellite cells specifically when we're talking about ganglion. So satellite cells, like in the dorsal root ganglion, they surround it and help to protect it and also do a couple of other jobs. So these are flattened cells that envelope neural cell bodies in the peripheral nervous system. They provide structural and metabolic support. They maintain the microenvironment in the ganglia, and they form a complete layer around cell bodies of neurons in the ganglia. So we saw this exact picture earlier. You have these satellite cells that surround the soma, or the cell body, also known as perikaryon. They surround it to protect it, give it the structure, make sure that everything in it is running okay. Again, these are our supporting cells. Next, Schwann cells. This is where we're going to dive into myelin formation. Remember, in the central nervous system, what were those cells called? Those are called oligodendrocytes in the central nervous system, and they myelinate multiple different axons. In the peripheral nervous system, and again, what's the difference between peripheral and central? Central nervous system is our brain and our spinal cord. The peripheral nervous system is going to be everything outside of that. The ganglia, the cranial nerves, you have all of our peripheral nerves going down into the muscles, our spinal nerves coming out of the spinal cord. All of those guys are part of our peripheral nervous system. So the Schwann cells cover the axons in the peripheral nervous system, and they cover all of the axons. Some of the Schwann cells, this is what we primarily talk about when we talk about Schwann cells, is that they can lay down myelin sheath. So it's a type of plasma membrane that's rich in lipid, which is why it's more white in color. A couple of things that we'll look at some of the anatomy. This should be Schmidt-Lantermann clefts, nodes of Ranvier, and internodal segments as well. Well, these are the big pieces of anatomy that we want to look at. So Schwann cells, another name for Schwann cells is neurolemocytes. I'm actually going to put that, aka excuse me. Apologies, my cats have been lonely today, so they've been rubbing on my desk and all over my stuff. So a little bit of allergies acting up. Okay, so the Schwann cells, aka those neurolemocytes. Right here we can see these Schwann cells. See how they kind of, as they produce this myelin, they wrap around it. There are some other vocab terms and stuff that we want to look at here. We'll look at that in a minute. So myelin is formed by the Schwann cells. It's also formed by the oligodendrocytes. In the PNS, it's the Schwann cells. In the CNS, it's the oligodendrocytes. I'm going to keep saying that because it is constantly tested. It's described as having a jelly roll fashion. This is what a jelly roll looks like. So kind of that wrapping motion. And one individual Schwann cell forms one segment of the myelin sheath. We call these the the nodes, these internodes. So one individual chunk of myelin. Each chunk of myelin is one single Schwann cell. Couple vocab, we have an outer collar of the perinuclear cytoplasm. That's called the neurolemma. There's a neurolemma, it's that outermost layer. We have myelin sheath gaps. Those are those nodes of, you can either call it Ranvier or Ranvier, however you want to pronounce it. They're gaps in the myelin. They're the nodes of Ranvier. And then we also have there are also non-myelinated fibers. Okay, here is some of the anatomy that we want to look at. So here are our Schwann cells. Each individual chunk of myelin has Schwann cells. Each of these portions of myelin we call internodal segments, and we call them internodal segments because of those nodes of Ranvier, which is the nodes are one of the most important parts of the axon. So we can see each of these portions are our nodes of Ranvier, and the myelin is what it does is it acts as an insulator. It insulates to allow, remember when we talked briefly about the job of the axons of the neurons is to transfer in a message. And the way that it does that is it passes the message along through the membrane of the axon. Okay? The axolemma, the membrane of the axon. What the myelin does is it allows us to kind of jump and skip parts of the axon so we don't have to fully do all of the work in each part of the axon. So it speeds up the process. This is going to be covered a lot more in the beginning of next trimester in Physiology 1, but I want to give you a solid base now. So let's say we have an unmyelinated axon right here, and the signal comes down. The way that the signal is passed is through what's called ion transfer or ion diffusion. So what it happens is it changes the electrical charge by letting certain ions in or out. So we have ions come out, we have ions go in, the charge changes, and then it continues. Ions go out, ions come in, the charge continues. And this is how it passes this message along, is through the transfer of ions. What the nodes do, or what these nodes of Ranvier allow us to do, and what the myelin allows us to do, is instead of having to do this all the way along the entire membrane, the myelin allows us to skip, to kind of jump. It's called saltatory conduction. Let me actually go back and add that in because that's a really, really good word to know. Schwann cells, myelin. The myelin allows for saltatory conduction. Saltatory, the Latin root saltar, salta is to jump, so it allows the jumping of it to happen. So see, so instead of having to come all the way down the entire membrane, it's letting ions in and out up to this point, and then it can skip this whole chunk. The ions flow, the signal travels here, and then ions flow in and out in the node, then it hits another myelin, so it allows it to skip whole chunks of the axon, so it doesn't have to do as much work, which allows the signal to travel much, much faster, which is, how we can immediately think about moving our fingers or moving whatever, and within milliseconds, we can do those movements, oftentimes without even having to think. Okay, so that's most of the anatomy, the important anatomy in the myelin sheath itself. This little video is really cool. This kind of shows the process of how the myelin sheath forms. So you can see, let it play. So you have the axon, you have the Schwann cell, and then it starts to wrap and form just like this, and you get those wrappings over and over and over and over again to protect the axon. So it insulates and it protects the axon. It's just another picture kind of showing that process. Another part within the myelin sheath, specifically within the PNS, and I should clarify, the myelination of this, it's very similar in the central nervous system, where it comes and it wraps just like this. The only difference is that instead of a Schwann cell, it's an oligodendrocyte off to the side, and it has a little arm that reaches out, and then this process occurs. Is because that one does, the oligodendrocytes do multiple axons all at once. So the clefts of Schmidt-Lantermann, these are only in the peripheral nervous system. They're small spiraling gaps within the myelin sheath that contain Schwann cell cytoplasm. They help to maintain and nourish the myelin layers. With this, what you really only need to know, you need to know the name of it, that it's maintains and nourishes the myelin layers, that's its job, and then be able to recognize it on a picture. A picture like this is going to come up most likely in the lab portion. Be like, hey, what's this? Most likely a picture just like this, or a picture just like this. This is what you're going to see, these little indents on the myelin sheath. And those are our clefts of Schmidt-Lantermann. Okay. Some of the differences, again right here, Schwann cells in the PNS, individual, whereas the CNS, you have the oligodendrocytes that go all over and do a bunch of different axons. But you do still have the same myelin sheath that wraps around. You still have the node of Ranvier, and all of those very similar structures. Always remember PNS, Schwann cell, CNS, oligodendrocyte. This is that process that I drew out that I was describing, where it jumps across. I'll actually add that term in here as well. Saltatory conduction, or jumping conduction. So the signal can jump. So that way not as much has to be done, and it can pass the message along much, much quicker. Those are the ions going in and out. Those are our internodes right there. Good. And here, showing just an extra little note that the peripheral nervous system axons are always covered by Schwann cells. They don't necessarily have the myelin sheath. So for example, this one does have the myelin sheath, so this is a myelinated axon. These are our unmyelinated axons, and this right here is a Schwann cell, and it's holding on to a bunch of different axons all together. So, but they are always covered by these Schwann cells. Okay, and that does it for nervous tissue. Thank you again so much for watching. If you have any questions, comments, concerns, drop a comment down below and let me know, and I will get back to you as soon as I can. Thanks for watching, and good luck studying.